First of all, thanks so much for the kind invitation. It's a beautiful city, and I look forward to talking more to you all. The talk here is one of two, as you have heard. In my talk, I will especially discuss how grid cells in the entorhinal cortex contribute to a mapping of external space, and then afterwards May-Britt will relate those cells to the hippocampus, place cells that O'Keefe discovered, and also contributions to memory. However, before I start, we'll also spend some time, a little bit of time, just to recapitulate the important findings of O'Keefe, since he couldn't be here himself. But before doing so, let me begin with a little bit of background. And let's go all the way back to, actually, how far to the side can I go? Not a lot. Just so I see if I see the slides. But let's go a little bit back in history, where May-Britt and I started some approximately 30 years ago. Because when we started to study psychology in the mid-1980s, we were interested in the neural basis of behavior. And our entry point was behaviorism. Behaviorists described relationships between stimuli and responses in the environment. And I can mention people like James Watson, Clark Hull, and B.F. Skinner. So this brought us into the field, and we found it exciting. I was very impressed by the scientific rigor, but clearly missed one element, and that was the underlying neural basis of behavior, which the behaviorists had deliberately avoided due to the lack of methods. So, at least, and another problem was also the fact that behaviorists like Skinner and so on, they describe behavior essentially in stimulus-response relationships. And that was also a bit simplistic. And then I want to mention E.C. Tolman, because he broke with that tradition. And with Tolman, a completely different view came. And what Tolman did was that he actually suggested that the brain has map-like structures, cognitive maps, as he referred to, which he used to account for behavior. What Tolman did was to study rats that walked in mazes like everyone else did. But what he discovered was that rats were able to take both shortcuts and detours. And why is that important? Well, the reason is that the behaviorists, the ones that I mentioned in the previous slide, if you go back to the more traditional behaviorists, is that they described behavior when a rat goes from one place to another. They described it as a series of stimuli and responses. So one stimulus led to the next, and this is so. This was not sufficient to explain actually how rats were able to take shortcuts and detours. So I will take one example. This is Tolman's famous sunburst experiment, where he started at one point and then over many trials of training learned to walk a certain route to a goal. And then on a test trial, the rat again walked to a certain place, and then number of routes were available that had not been available during training. And what Tolman observed was that the rat actually on this test trial, instead of walking the route that it had always chosen during training, took the direct route that had never been available previously. Why is that important? Well, it sounds like a trivial result, but it is actually extremely important, because it meant that the rat could not get from one place to another just by linking a long series of stimuli responses. the way Tolman explained this was that the rat actually had an internal map that led it from one place to another. This is the only way that he could explain that it could take a new way from A to B. Of course, this experiment has been criticized heavily for methodological reasons, but I will not go into that. The important thing here is that Tolman introduced new concept of a map-like representation of the external world in the brain. But Tolman himself was just like the previous behaviorists, not interested, at least not in his early days, in explaining behavior with the underlying neural basis. So he stayed away, and like most others, in contrast to just a few others, like Lashley, who actually started the field of physiological psychology by trying to explain learning and memory by the underlying neural basis. And then Lashley's student, Donald Hebb, was the one who really conceptually linked physiology and psychology by introducing concepts that excited, concepts like cell assemblies and plasticity, that excited a whole new generation of researchers who actually wanted to go into the basis of behavior, which the previous behaviorists had not dared to do. So this led to gradually build up the basis for some excitement in the field, and even Tolman himself, at the end of his life in 1958, expressed that he thought that this physiologizing, as he called it, the attempt to explain mechanisms of behavior, was where the new breakthroughs would come in the future. So this was around in the early 90s, or in the end of the 1950s. But at the same time, there was another important development, and that came from the other side, not from physiology, but not from psychology, but from physiology, where two pioneers of the field in physiology, David Hubel and Torsten Wiesel. Torsten Wiesel had then moved from Sweden to the United States and worked with David Hubel, and what they did was that they actually began to record activity in the cortex, in the visual cortex, specifically to try to understand how the brain generated visual images. And as you all, or many of you probably know, they found that the visual image could actually be broken down into elements like that were represented in individual neurons. For example, individual neurons might respond to edges, lines, and so on. So this led to enormous advances during the next decades in the understanding of how the visual image is represented in the brain. It had enormous implications for perception, also in other modalities, and it even led to the development of a field of cortical computation, where people had enough data to actually try to model computational processes in cortical networks. However, their work was confined to the low end of the visual cortical hierarchy, and I try to illustrate that by this image from Van Essen, which shows the areas of the brain that are involved in vision. And only down here you find V1, which is the area where almost everything has been done in visual neuroscience, of course with some exceptions, but much of the work has really been focused on the low end here. So as we go further up, things get much more difficult, and we get fewer and fewer examples where we actually are able to relate a property of the visual image or the external world on one hand to a clear property of neural activity on the other. So correlations to the external visual world get rare as we go up in the hierarchy into the visual system. And the same can be said for other sensory systems. As you go away from the primary sensory areas, you get in neural activity gets much more difficult to interpret. However, yeah, this is just to make that point, but if you now go further up to the real top of this cortical hierarchy, then there is actually one exception, and that exception is the place cells that were discovered by John O'Keefe. And I'll show you one example of such a place cell by I'm going to show you a rat. A rat is walking in a square box 1 by 1 meter and chasing chocolate pieces that are scattered around. And while the rat is doing that, we are recording activity from one single cell in the hippocampus. We're going to listen to that cell so that whenever the cell fires an action potential, there is a sound. So let's now see if the sound works here. Oh. Well, it's hard to... Oh. Okay. So what you see here is where whenever you hear a spike or whenever there is a dot here, the cell fires. And what you can see is that the cell fires whenever the rat is in this upper left corner here. That's an example of a place cell. The cell only fires when the rat is in a certain set of locations. Yeah, it's okay, I'm done. Yeah. No, there's no more. Yeah. No, there's no more sound, so no worry. Okay, so let's see here. A place cell is shown here. So this is a way to represent this activity. So red color shows high activity and blue shows low activity. And this is the box. So you can see that this particular cell is really firing only when the rat is up in the left corner there. So O'Keefe found many such cells in the hippocampus, and together with Lynn Nadell, he then in 1978 suggested that these cells form an internal map because every cell has a different preferred firing location, so that together all these cells form the activity of all these cells can tell anyone, including the brain itself, where the rat is at any given time. So therefore, O'Keefe and Nadell suggested that these place cells are actually part of the cognitive map that Tolman suggested in his maze experiments. So then, May-Britt and I, we went to London and we worked with O'Keefe for a few months, and then went to Trondheim to set up our own lab. And one of the questions that were important was, of course, where does this signal come from and how is it generated? So this is one of the things that we asked when we came to our own lab. And I'll show you where the place cells were recorded. This is the hippocampus of a rodent brain. This is a transection through the hippocampus. Looks like this at all levels of the hippocampus. And CA1 is this area, the last stage of the hippocampus, where most place cells had been recorded by that time. But hippocampus consists of several stages, and it works more or less like a unidirectional loop, starting here with the entorhinal cortex, which provides most of the cortical input to the hippocampus. Then we have the areas dentate gyrus here and CA3 here, and we have the CA1 here. And the flow information is essentially unidirectional, so it goes out here and then it actually comes back to the entorhinal cortex here, although that is not shown here. So a common idea at that time was that the place cell signal was generated in the hippocampus and sort of built up as information flows through the circuit. However, that might not be the only explanation, because there are also direct inputs from the outside. They were kind of ignored for many, many years, but there are direct inputs from the entorhinal cortex to CA1, to the CA3, and of course to the dentate gyrus. And they might also contribute in this processing. So the question we asked together with our student Vegard Brun and our collaborator Menno Witter was whether it actually is, if we record activity from the CA1 of the hippocampus, and if we interrupt the flow of information through the hippocampus, whether we then would lose the place signal. That should be a prediction if the signal is actually made in the hippocampal circuit. Alternatively, if the hippocampus receives the information about place from the outside, for example through this red input here, this should not matter all that much. I'll show you what we found. That's shown here. Let's first see, here are just a verification of the lesions. So you see the hippocampus here, and then you see the area that is blocked here, and here, and here, and then this is where the electrodes were so that we could pick up the signals, and then the result is shown here, 7 different cells, and you can see that they all have strongly preferred firing location. So this cell fires only when the rat is up here, this cell fires in the middle, this cell fires in the top left corner, and so on. Perhaps they are a little bit more blurred than they used to be, but not a lot. So, because the place cell signal still exists, it implies that perhaps there is more that's coming in from the outside than people had expected. So for that reason, we then decided that we wanted to go to the entorhinal cortex, to the cortical input region, and actually record activity there. And this is work that we did with Marianne Fien, Sturla Molden, and still with Menno Witter. And this shows the entorhinal cortex. It's a rat brain seen from behind. This is the brainstem, this is the cortex, this is the entorhinal cortex. And our recordings were made in the dorsal part because the dorsal part projects directly to the area where most place cells had been recorded by O'Keefe and others. And I'll show you a video now without sound, so just relax. And we'll see where the cell, a typical cell in the entorhinal cortex, fires. So again, the rat is walking in this square environment. Each white dot here indicates a location where this single cell is active. And you can see that it is not as clustered as it was in the hippocampus. So now we speed it up just to see it better. And you can see that the cell fires all over, more or less. However, if you watch for a little while, you will see that there are actually discrete firing locations. So the cell does have spatial activity, but there are many firing fields, not 1 or 2 like it usually is in the hippocampus. And the other thing you may observe is that there is some peculiar pattern here because the distance between each of these firing fields is more regular than you would expect by chance. So we followed up this, and now we're Torkel Hafting also on board. And this is a typical example of a cell that is recorded in this area. So here you have a box that is much larger, 220 centimeters by 220 centimeters. The gray trace is the path of the animal as it has been walking for approximately half an hour. And each black dot is the location where a spike has been discharged. So you can now see that there are many firing fields, and they do really form a regular pattern, and this regular pattern we refer to as a grid. The pattern can also be seen for this cell in the bottom left here. So you see, again, the black dots on top of the gray trace, but now I try to put lines on top of it just to help you see that the underlying pattern is actually triangular, or hexagonal, if you like, grid that repeats itself all over the available space. So because this grid pattern is so striking, we called the cells grid cells. So then we found that there are actually many grid cells in this area of the entorhinal cortex. They come in different varieties in layer 2 of entorhinal cortex. Most of the cells, or at least half of them, not all, but many of them, are grid cells. But they differ in several ways. And there are at least 3 dimensions of variation. I mentioned them here. First, there is the phase of the grid. That means that if two grid cells have different phase, it means that they are shifted in the XY plane. So you see a blue grid cell here and a green grid cell, and they are located, they are shifted in the XY direction. So that means they have a different phase. Or we can also find cells that have different scales. That means that the cells have different sized firing fields, and the distance between the fields is different. So illustrated here with a green cell and a blue cell, and you can see that the blue cell has a larger scale than the green cell. Or the orientation of the grid may differ. So it's illustrated here by different degrees of tilt. So the green cell has an axis that is illustrated by the black trace here, and the blue has an axis that's illustrated by the red trace. So several dimensions of variation. So we thought that perhaps these dimensions of variations are somehow topographically mapped in the entorhinal cortex. So let's take a look at one at a time and begin with the phase of the grid. Is the phase of the grid topographically mapped? So what we did was to record activity from cells that are very close to each other using these tetrodes. They look very big, but they're actually just 17 micrometers each. So they are so small that they actually are about the size of a stellate cell in the entorhinal cortex. A stellate cell is approximately 20 micrometers across the soma. So, but they pick up activity from neighboring cells. And then let's look at some cells that are recorded on the same electrodes. So first the blue cell, and then another cell that was recorded simultaneously from the same tetrodes, illustrated here by red, and another one that's illustrated here by green. And what you can see is that the 3 cells have different phases. They are not located on top of each other, but actually shifted in the XY direction. So that illustrates the point that the organization of phase, I mean the exact location where the cells fire, is actually quite random. It's completely random within the resolution that this technique allows for. Often this is called a salt and pepper organization in other fields, and why it is called that is actually, it just means that this is what you get if you have salt and pepper and mix them as much as you can. So this is the same here. It's non-topographic, completely non-topographic. So that means that at any given location in the network, you will have cells that have all possible places represented, which then means that this is a very efficient way to make sure that at every possible place in the anatomical network, every possible location is present. So how about the scale of the grid, the second parameter of variation? So here we have a rat brain, sagittal section. This is the entorhinal cortex. This is the hippocampus. And what we found is that as you start from the top, from the dorsal end here, and go down towards the ventral end, the scale of the grid on average increases from small meaning approximately 30 centimeters between each of the fields, to very large, meaning several meters between each of the fields. This is a bit difficult to measure, so we had boxes that were up to 220 centimeters by 220, in one case actually 400 by 400, but it gets very hard to record when space grows so much. So we did recordings in linear environments and then extrapolated, and I think we can say that the scale really increases to at least 5-10 meters distance in the ventral parts. So here we have really a topographical organization in the sense that the scale increases as you go down. It's also shown here in the early data as we averaged across animals. This is what we saw. Scale or the spacing of the grid increased as you go from dorsal to ventral. But it's not completely true that this is something we see in average data. But if you go to individual animals, the story is a bit different. And this is more recent data from 2012 with Tornhan Stensola, where all the data shown here now is from one animal. Each dot here is one cell, and this is dorsal to ventral position as before, and on the y-axis as before, the spacing of the grid. So you can see that there is an increase in the scale of the grid as you go from dorsal to ventral. But you can also see that it's a step-like process, so that there is not every possible spacing is present. There's 4 discrete steps, and this is present. This you can see in every single animal. There's no exception. They always have these modules, as we call them, where cells sort of, as you go from dorsal, you have the smallest one, then you recruit another level, but you don't drop the original level. Then you recruit yet another level, and as you get down approximately halfway, you get the 4th level, sometimes the 5th level, and further down we can't go because the spacing gets too large. But a discrete modular organization of grid spacing, and then, of course, it becomes natural to ask, how are these modules organized then? So let's call the small one M1, the next one M2, M3, and M4. If we now divide M2 by M1, M3 by M2, M4 by M3, we can ask if there is a certain scale factor. And that's actually the case. So as you see here, this shows the scale ratio. That means M2 by 1, 3 by 2, or 4 divided by 3. For 32 modules from 11 rats, so each circle is one module, and the crosses are the averages. And what you can see is that the averages show very little variation, actually is around 1.42 on average. So that means that as you go from the smallest module here to the next, you just multiply the scale by 1.42, you get the scale of the next, then you multiply again by 1.42, and you get the scale of the subsequent one, and to get the subsequent one, the one after, you again multiply by 1.42. So this is like a geometric progression where you just get the next element simply by multiplying by a constant factor, 1.42 or square root of 2 if you... you can ask about that afterwards, but it's surprisingly close to the square root of 2, which is 1.41 something. So what's the advantage of organizing the grid map in this way? Well, we don't have experimental data on that, but theoretical physicists have worked on it, both the Matti of the Hertz group in Munich and the Balasubramanian group at UPenn. And what they both find is that this is the optimal way of organizing the grid map, in the sense that it is the way that allows the brain or the entorhinal cortex to represent the environment at the highest possible resolution with a minimum number of cells. So it's economical because you can get maximal resolution with as few cells as possible. So then the third factor, the orientation of the grid. I'll show 3 examples. You can see that these cells recorded at the same time in the same animal show different orientations. So it's not one single orientation. You can see they are tilted differently. It's also shown here. And if you then compare, let's just focus on the left one. So if we now ask, is there any relationship between the orientation and the spacing, and then plot those for a number of simultaneously recorded cells. So each cell is shown by one circle, and you can then see that this particular cell, for example, here has an orientation of approximately minus 50 degrees, and a spacing of approximately 30 or 40 centimeters. And then you do the same for every single cell, And what you can see is that there are an obvious clustering here, meaning that those cells that have the smallest spacing tend to have similar orientation. Those that have the next spacing tend to have similar orientation, and same for the next level, and then harder to say for the highest levels because there are so few cells. That means then that grid spacing and grid orientation are co-modular. That means that they shift at this, they have the same boundaries. The same can be said for a number of other properties of the grid. They even respond, different modules may respond differently to changes in the environment. So it's really a module that not only is described by differences in spacing, but they actually behave differently vis-à-vis the external environment. So finally, in this introductory part, let me just mention that the grid cells within modules are extremely rigid. That is illustrated here by this cartoon. So again, the three cells that were recorded more or less at the same location, illustrated by different colors here, or shown down here. So you have the blue grid points, grid fields here, the red ones to the right, and the green one below the red and the blue. And then what happens if you go to a different room, for example? Again, you maintain the same pattern. You see here, blue, red, and green. And again, as you go back. So the point here is that this is one single map that is applied on the environment in the same way, wherever you are. So cells that belong to the same module in the grid map, they are really stick together so that those that fire in the same location in one environment also fire at the same location in another environment. So this is very different from the hippocampus with the place cells, as you will hear in May-Britt's lecture, because the place cells, they make unique maps for every single environment, and that's exactly what you want for a map that is also used for memory. Whereas this map is more useful for expressing relationships in space, including distances and directions. It's like a ruler or a metric that you really don't want to change that for between environments. So if you go from one room to another room, you want the distances to be the same. Otherwise, if you have to generate a new metric for every single environment, you would run out of capacity very soon. So let us, I'll skip the data here because this is just too complicated. But then let me just sum up the introduction by letting you know that the grid cells are really not only present in rodents. So began with rats, and then we found them in mice very soon after. Not so surprising. But then rats and mice are very close on the rodent branch. But then in 2011, Nachum Ulanovsky's group found them in bats, in Egyptian fruit bats, and they are on a completely different branch. And then they were found in monkeys in 2012, and then in humans in 2013. So grid cells exist all over, or at least across widely separated branches of the mammalian evolutionary tree, which then suggests that they must have arisen at a quite early stage of mammalian development, or even possibly before. Okay, so in the second part of the talk, I want to address three questions that we are working on at the moment. One that is unpublished data, that is just will come out in a month or two, and then I will also address some questions, some even unfinished work, and point to the future in the final part. But let's begin with the first part, which is where I really want to ask how does the grid align with the environment? What I mean by that. Do you remember the grid had certain firing locations in a box? And then we can ask, these firing locations, if they are used for the rat to actually know where it is, they have to be in the same locations every single time. So when the rat comes back to the box a second time, the cell has to fire in the same spots then as it did the first time. Otherwise, it will not be very useful for navigation because the relationships will be different. So what are the rules that make cells fire at certain places and not other places? So one of the observations that were quite intriguing is shown here. So this is a set of data from 4 different rats, 4 different grid cells. You can see they have these hexagonally spaced patterns. And what you see up here is the gray trace is the part of the animal, and then the black is the spike locations as before. In the bottom row here, you see plots for the same cells, but they are, these are what we call autocorrelograms. And what is that? That is just a kind of average of the spatial organization of this map. So it shows the average location of fields relative to other fields. So this is a kind of average of the grid pattern here, and this is an average of that, and this is an average of that, and so on. What you can see is that the orientation of the grid, how they are tilted relative to the box axis, is actually surprisingly similar in all. And this is just 4 different examples. So what we wondered, together with Tor and Hans Stensula, who did most of the work, was that whether this was really present in all cells, were they so similar in orientation, or what were the rules? So what we did was to again use the autocorrelogram for each cell and then plot the inner 7 points of the autocorrelogram. So the reference point here in the center, and then the 6 surrounding points like this, and then just transfer those 7 points to a diagram of the box here. So this is the recording box that you have seen so many times now. The green is the x-axis of the box, and the orange or the brown is the y-axis. And then we take the 7 points of the axis, put them on like this, and then do the same for all other cells. So now we have 7 points for every single cell out of a pool of 587 cells. And what you can see now is that this is certainly not random. So it's not uniform distribution, but the cells actually tend to line up along the x-axis, which is the green axis here, and 60-degree multiples of that axis. So the green here, and the green there. And they tend to avoid the y-axis here. You see there's not a single cell that has firing locations that line up with the y-axis or 60-degree multiples of it. That's also just shown here in the histogram. So this is frequency on the y-axis, and then orientation of the grid on the x-axis. And then you can see that they sort of line up around 0, which is the horizontal axis, and then 60-degree multiples of that. However, if you now look more closely here, so let's take one example. You have an autocorrelation map here, and then you can see that this is an example cell where you have an angle between the axis of the grid, which is shown here, the top line, and the horizontal reference line that corresponds to the x-axis here. And then we measure that angle. So we measure the angle for the axis that is closest to any of the walls. Then we plot it again, same as we did on the previous slide. So number of cells on the y-axis, and then the orientation of the grid on the x-axis. And what you now can see, if you now focus in, you actually even saw it a little bit in the previous slide here, is that it doesn't really perfectly line up with the x-axis, but there is some tendency for cells to cluster at either a certain positive shift away from the x-axis or a certain negative shift. So 0 degrees, meaning perfect alignment with the x-axis, almost doesn't exist. And then there are many, many cells that cluster around approximately 7.5 degrees away from the wall axis. And then almost none that have orientations that are close to 15 degrees. Why is that? The cells tend to shy away from both 0 degrees and 15 degrees and really cluster around 7.5 degrees. So a possible explanation is shown here. So now we plotted a triangle to illustrate the grid for 0 degrees, for 15 degrees, and for 7.5 degrees. So what can be said about 0 degrees and 15 degrees is that they actually have symmetry axes that correspond to the symmetry axis of the box. So one symmetry axis of the box is the vertical axis here, because it's similar to the walls here. And, and another one is the x-axis here. And you can see that the yellow grid has one axis that is identical to the box axis here. And the same here, the yellow axis, the yellow triangle here is symmetric around the diagonal, and the diagonal is one of the symmetry axes of the box. And this is in contrast to 7.5 degrees, which is as far away as you can get the grid from any of the symmetry axes of the ball, any of the wall axes. So in other words, at 7.5 degrees offset, the grid is as dissimilar along the x-axis as along the y-axis. So this is the way you can orient the grid to make it as different as possible when you walk along the one axis or the other axis of the walls. And this, although we don't have experimental data for it, this suggests that the orientation of the grid is actually such that it makes the total activity of the network as different as possible in parts of the box that are physically similar. So let's then ask, how can this come about? And if we then again go back to these polar plots that show how the different cells are clustered relative to each other, then let's compare the three axes. So now, until now, I just talked about the axis that is closest to one of the wall axes, and I said there is this 7.5 degree offset of the grid, and in this data 7.9 degrees. But if you look at the other axes, those that are more offset from any of the walls, you can actually see that they are not shifted as much. You can see it here, and you can see it here, or quantitatively here. This is the frequency of cells, and this is the degree of offset. This is the axis that is closest to any of the walls, and this is 7.9 degrees, as I said. But the other two have smaller offsets, 4.4 and 2.6. And what does that mean? It means that if you have three grid axes and they are shifted to different degrees, then it must imply that you have lost the hexagonal symmetry, because there's no hexagonal symmetry if you shift the axes in different amounts. So that means you have got an elliptic grid. Elliptic in the sense that the inner parts of the field here are not circular anymore, but actually become elongated, like you can see here, because the axes have been shifted differentially. And actually, there is even a correlation between the offset of the grid axis and how elliptic the grid gets. So that suggested to us that there might be an underlying process that is quite similar to what is called shearing in mechanics, in continuum mechanics. So shearing, that's a type of deformation of any pattern that is characterized by a shift that is not similar all over the surface of the object, but actually becomes less pronounced as you get away from the walls or from the axis. So to illustrate what I mean by that, let's take a grid pattern here. You see a hexagonal grid pattern. This is just simulated data. And then I indicate the shape of it by a circle here in the center. Then let's introduce these forces, which we can call shear forces. So now we have an axis, and this is the y-axis in this case. And then there are forces that are strong, close to the walls, close to the right wall here, or to the left wall. In this case, they go in different directions. And then the strength of those forces decrease as you get away from the walls at either side. And this is what characterizes shearing, that you actually have forces that decrease as you get away from the shear axis. So what happens if you have such forces is described by this function here. Well, what you get is, if you shear this pattern here, you actually first you rotate it, as you can imagine, because of the direction of the forces. But in addition, you also obtain an elliptical deformation of the grid at the same time as you get this rotation here. So that means that you can get the elliptification of the grid, or the deformation, and the rotation of the grid by just introducing forces that can be described by very simple transformation that actually works for many phenomena in nature, not only for grid cells. So knowing that you can actually get these deformations of the grid pattern by these shearing forces, we then applied those forces on our data, but in the reverse direction of what we thought had happened. So again, let's look at those frequency plots here. And you have the three axes here. Let's focus on the axis that's closest to the walls. This is the original data. You remember this is peak at 7.5 and minus 7.5 degrees. And then if you apply these shearing forces that are close on top on the grid pattern, forces that are really strong near the walls and then decrease as you get away from the walls, you can see that you completely remove the 7.5 degree effect. If you do it in the other direction, you have no effect. So this is really direction specific. So to sum up that part, I'll show you I'll show you an example that may illustrate the forces that apply on the grid. So here you have a stack of cards, and here on the side of the stack, Tor Stensola, who made the illustration here, has drawn a grid pattern, or the three axes of the grid, and then one of the axes is shown in black. That's the axis that is orthogonal, or 90 degrees compared to the forces that he'll apply. And the forces can be seen here. So you have white arrows here indicating the strength of the forces. So they all are in the horizontal direction, but they decrease as you get away from the top surface. So now let's apply those forces and see what happens to the grid pattern on the stack. Let's see. Okay, once more. That was a bit. Oh, där uppe. Okay. I can only see it there, but it's fine. I'll do it like this. Okay, so look at the stack and at the grid pattern. You see? So this is what we imagine actually happens on the cells. So because of these forces along the walls, then you actually get the deformation of the grid. And you see that it's also rotated so that the axes are shifted away from the walls. So how this happens, how these forces are actually applied on the grid pattern, that is still to be determined, but it's quite likely that it involves cells that specifically represent the borders of the environment, which you will hear more about in May-Britt's talk. So that suggests then that the grid pattern interacts strongly with the local boundaries of the environment, the walls in our case, and that they sort of, in the process where grid cells are anchored to the external environment, there are these forces from the environment actually adapt or deform the grid pattern as the animal experiences the environment. And data that I have not time to show you will suggest that this, when the rat is exposed to the environment for the first time, it is actually a very symmetric, nice pattern, and then over time it gets rotated and shifted as the animal gets more experience. So the second data I want to show you goes back to the original point I made about the anatomical organization of the face of the grid, where the grid fields are located relative to each other. So what I suggested, based on the tetrode recordings, or the electrode recordings, was that there is no clear pattern of the grid face in the local network. However, we also know that there is a limitation to tetrodes, because each electrode here, or tetrode if you want, picks up activity from a distance of approximately 50 to 100 micrometers around the surface area where the cells are, where the activity is recorded. So within that area of 50 to 100 micrometers, we can't really tell where a signal comes from. It may come from the blue cell or the green cell or the red cell, but we don't know how they're located relative to each other. So we need better resolution. And how can we obtain that? Well, that brings me to a new method, which is two-photon fluorescent calcium imaging, which has been used in other fields of neuroscience for a few years, but only on superficial parts of the brain, such as the visual cortex. It has allowed researchers to actually study activity in a map of cells as the animal is performing behavior. So, for example, when an animal is looking at a visual image, people have been able to visualize activity of maybe 100 cells, maybe several hundred cells at the same time in parallel with behavior. So we try to apply this to the entorhinal cortex. And this is then a collaboration with Tobias Bonhoeffer at the Max Planck Institute in Munich, and Albert Sau, our graduate student, has spent a year there to do these experiments. So I'll then suggest, well, actually, let me first take the problem here. The problem with the entorhinal cortex, if you really want to image activity in the entorhinal cortex, the problem is that the entorhinal cortex, as you saw on previous slides, is really located at an almost impossible location. Because here you have the brain, the cortex here, the cerebellum here, and the entorhinal cortex is at the back end of the cortex, close or adjacent to the cerebellum, and lying underneath the transverse sinus, so that which contains a lot of blood, and you really don't want to break that. So how to get the microscope in here is really a challenge. But it is possible to do it, and what we have done together with the Bonhoeffer group is to introduce a prism that goes into this cleft here, a very, very small prism that goes into the cleft as illustrated here. So here you have the prism, of course much, much smaller than indicated here. And then it goes down between the cerebellum and the cerebral cortex. And then the back end of the entorhinal cortex here can be imaged from the top of the prism here. And you apply a lens on top of it here, and you can actually see the back end of the entorhinal cortex. And it's beautifully intact because the prism is just between the cortex here and the cerebellum without damaging any of them. So when these prisms are inserted, the animal is under the two-photon microscope. We can then inject a virus into the mouse brain, and that virus contains then GCAMP6, which is a fluorescent marker, calcium marker that is expressed in cells, and when 3 weeks or so have passed, then the cells in the area, in the entorhinal cortex, express this calcium indicator, and we can then put the animals under the microscope. And then, at the same time as we look at the cells, we then let them run in a virtual environment. So animal, the mouse, is located here, running forward on a ball, doesn't move, but then its movements on the ball on which it is moving, then is translated to movement in this virtual environment. I can play it once more. So as the animal is moving forward on this stationary ball, it is actually environment is projected onto a screen here in proportion to the distance that the mouse moves on the board. So it's really like navigating in a game, in a computer game, and this allows us to image activity while the mouse is completely still, it's head fixed on the board here, and we can then ask if the grid cells in this virtual environment represent space. So this is now the entorhinal cortex seen from the back, and 6 different imaging windows, imaged one at a time. If you now look at the top one here, which is magnified, you can now see the activity of different cells lighting up in white here as the animal is moving from left to right on this virtual track. You can see how the cells, they fire only at certain locations, each of them, meaning that the cells are actually spatial and possibly grid cells. So if we then go to illustration of this activity, so this shows now the track here, 8 meters long, the virtual track, and then different runs. This is the same cell, and activity, red shows high activity, blue shows low activity. And what you can see from this cell is that it tends to fire at the same locations, and those locations are periodically spaced. There's a regular distance between them, which really suggests there are grid cells. But remember, this is just one dimension. It's just a track through two-dimensional space. When the animal goes in the other direction, this is the return path, then you can also see that the cell fires in certain locations, but it is less regular. And that brings up a problem, because when the animal runs in one direction, which is all it currently can do in this virtual environment, it's not obvious that the direction through which if it were a grid cell, the direction through which the animal would run would actually correspond to one of the axes of the grid. It may or may not be the case. So here are 3 different examples, which are certainly possible directions through a two-dimensional space. You can see that they may not at all correspond to the axis of the grid pattern. But what we did then was that we compared the actual trace here with a simulated grid that where we took a one-dimensional slice through a two-dimensional environment in all possible directions, at all possible locations, and even at all possible ellipticities in this grid, and then found the pattern that most closely matched the observed data. And you can see here that there are, if you just search the entire space of orientations and directions and locations, you will find some that really, really match the activity as a function of position. So this is a method that was introduced by David Tank already 1 or 2 years ago, but it works actually quite well for finding out which cells are grid cells and which are not. So now we have a way to find grid cells, and then we can ask, how are the grid cells organized in anatomical space? This is one of the windows that I showed in this previous slide. I can go back just to show you. It's one of these windows, or one of these windows. And we then asked, how are grid cells within these windows organized in space? Okay, so here's one such window. And the cells that pass the criteria for grid cells are shown in blue. The gray cells are other cells that do not look like grid cells. So then we try to measure, is there any clustering of the grid cells in this window? So we took a window of, for example, 100 micrometers in diameter, and then ran that window across the entire space, like this. And for each step, we counted the number of grid cells in the window, and then plotted it. So the observed data in this window is shown here. So you have frequency, number of cells in each window, here. And then, so number of cells in window is on the x-axis, and then frequency is the number of instances. How often you actually encounter the window that, for example, has 10 cells within the window. And of course, 10 is really a lot. But you can see that the distribution is quite wide, and there are quite a number of windows that actually has a number of grid cells in them. And now we compare that with a shuffle distribution. That's the red one. A shuffle is random. So then we have just taken all the grid cells and scattered them among all possible cells here in a random manner, done that 1,000 times, and then get this distribution here. So the take-home message from this distribution is that there are actually that the cells are actually more clustered than you would expect by chance, because there are many windows where there are many grid cells together, so which you would not get just by chance. So this suggests that the grid cells are organized in clusters, so you have local groups of grid cells, and that brings up then the question, if there are such clusters, are they more similar than others? So is there any organization, functional organization within those windows of grid cells? And this is still ongoing work, so I can't even give you the answer, but there are examples where you can see that those clusters really have similar orientation, for example. So here we just have taken the 3 axes of the grid for each of the cells that are inside the window, and then plotted them on top of each other. And you can see this is not random. There is a preference for certain orientations. And finally, back to the question that I started out with, within, if you now look at grid cells that are really close to each other, do they have a more similar grid phase than what we would expect by chance? And this shows spatial correlation, that means how similar they are. And then this shows the distance between the cells, between pairs of cells. And you can see that if you're down at approximately 50 micrometers distance, which is approximately 2 to 3 cells width, then cells start to get very much more similar than those that are further apart. So that means that the data that we initially recorded with tetrodes, with electrodes, suggests that overall there is a salt and pepper organization, and it's pretty random. But if you really go to the microscopic scale, there is similarity between cells that are very close, which again has implications for how the grid cell network works. And that brings me to the final 3 slides, where I will just ask one important question, where I don't really have the answer. This is a work that is more for the future, but it's one of the most important questions, namely, how does this grid pattern come about? So the grid pattern is not imported from the outside. There's nothing in the sensory inputs that looks like a grid pattern. So this is made up by the brain itself. So how can it be made? And then, this is too complicated to really, but I still just want to give you a feeling of what could be the mechanism. And then I would like to introduce the concept of continuous attractors. So attractors is a concept that comes from theoretical physics, and it refers to the fact that a neural network may have certain states, and then it may switch between those states. And when it's continuous, it may go from one state to another state in a smooth manner. So that's the general way to describe it. But let's now try to be a bit more specific. So let's imagine here we have a neural network. Each cell is one place cell, for example, or it could be one grid cell. But let's begin by place cells. And those place cells are organized according to where in the box they fire. So those that fire in the top left box are put here. Those that fire at the bottom right are put here. Then we add connections between cells. So those are the red lines here. So these are excitatory connections. And the idea is that activity in such a network is maintained by connections between cells that have similar firing properties, similar firing locations or similar place fields. So you can actually self-maintain activity in such a network if you have cells that mutually excite each other. And then the idea is that this activity is updated according to where the rat is in the external environment by signals that tell the network about both the direction that the animal is moving and the speed at which it is moving. So when the animal is moving from one place to another, it's reflected by movement of this activity bump in the network. So when a rat walks like this, the activity bump moves like this. And of course, cells can't mutually excite each other without having a ring of inhibition around it. So that's why, because otherwise it will get completely out of control. And that's why this is really called a Mexican hat type of network. And now a challenge has been that Well, the first challenge I want to introduce is that where do you get the hexagonal pattern? And that's quite easy, because you may have such patterns many places. You may have such excitatory connections many places in the network. If you have mutual excitation with inhibition around many places in the network, they will mutually inhibit each other. And if you have many points that all push each other away, they all inhibit each other, the only stable pattern that exists is hexagonal, because if everyone avoids everyone, it has to become a hexagonal pattern, because this is the way they're really as far away from each other as possible. And this is illustrated here by the Mexican hats that sort of fall into a hexagonal pattern. But then there was this challenge that actually we know from collaborations with Menno Witter and from others that there isn't much excitation in the grid cell network. And then Jasser Rudi, who is a group leader at our institute, found out that you can even do this just by having inhibitory surrounds at each cell. And in that case, because it's no longer a Mexican hat, then it's we call it an inverted Lincoln hat. So you can see that the point is just that if everyone inhibits everyone, you actually get a grid pattern. So I'll show you very quickly a simulation here, which shows that you start out with an activity that is random in the network, and then in the computer, after a very short while, it's all self-organizes into a beautiful hexagonal pattern. So this suggests that that hexagonal activity may actually be the result of competitive interactions in the network. Cells compete with each other and end up with this pattern. This is most stable of them all. And then this pattern can be moved around in accordance with the animal's own movement in the environment. And as long as this is continuously updated as the rat moves around, you actually get a hexagonal pattern in individual cells. And that brings me back to the summary. I'm not going to repeat things point by point, but I want to go back to Tolman, which is the one who introduced the cognitive maps or the spatial maps and suggested that there are such maps inside the brain. He did not have the tools to investigate such maps, and he even refused from speculating about the neural basis of these maps, except that at the end of his life, when Donald Hebb came with his ideas about how cells could actually collaborate or cooperate to generate complex or higher brain functions, then he saw the potential and actually suggested that this is where the advances would occur. So now it's more than 50 years later, and we have learned a lot about the neural basis of behavior, and we even are at a point where we actually see that patterns or patterns that actually apply also in other areas of natural sciences, such as physics. This is an example from a superconductor, where you also have repulsive forces that actually generate a hexagonal pattern of current vortices that is very, very similar to the proposed mechanism for grid cells. This just illustrates, together with the effects of shearing, which I've taken from paleontology here, where you see a trilobite that has been exposed to similar forces, where you get these distortions of patterns because layers shift differentially. It just illustrates the fact that we are at the point where neuroscience is developing both into a quantitative science and a science that actually uses principles that we can see throughout nature and not only in the brain. So that will be exciting to watch, and I think what I now want to do is, instead of acknowledging again all the people, I will leave that to May-Britt because she's going to do it at the end, but I want to acknowledge some of the participants who are not on the traditional acknowledgement slides. So with that, I just thank you a lot for your attention. Yes. So the grid cells are in an area of the brain that is among the very first to be affected in most cases of Alzheimer's disease. So as you know, Alzheimer's disease starts usually with a loss of the ability to navigate or to orient oneself in space. And this is something that happens usually long before a diagnosis is made. So there is a very likely link because Alzheimer usually starts in the entorhinal cortices. But there has to be additional work, of course, to link it specifically to grid cells. But that said, grid cells is the main cell type in the medial entorhinal cortex, so that it's very likely that there is a connection. Yeah, you mean how stable are the cells? Is that what the question? Yes, they are extremely stable. So when the first time an rat is exposed to a new environment, of course it takes a few minutes before things settle. But after that, it is so stable that you actually, if you have an angle of 7.0 degrees, for example, in one experiment, it will probably be 7.0 the next time as well. So, and this lasts for as long as we are able to record the cells, in some cases several months. So it's, this is really what you really want if you want to have an exact map that can measure distances and directions. So yes, it's extremely stable.
I think we have one more question.
Once more, if you would have. Oh, yeah. Well, it probably is. There has been one set of experiment where place cells were actually recorded in a spaceship. So the rats were, this is about 15 years ago, they were sent out in space, so they were all weightless and sort of navigated mazes that had all kinds of directions. But not so much came out of that experiment. But that said, if we forget that experiment, it's still very likely that vestibular senses are really important for grid cells in a general sense. So we see that when the mice are head fixed and they navigate in that virtual environment, then we do have grid cells as long as they're allowed to move straight forward. But once you actually extend the virtual environment to 2 dimensions, so they just navigate on the surface, then there is a mismatch between what they see by their eyes and the actual lack of movement of the head axis. So it just suggests that, together with many other observations, that vestibular inputs are important, and I think this would be messed up quite a lot under lack of gravity.
Okay, thank you. We have one more. We have some computer problems.
My question is, all this is about visual orientation, more or less. If you compare with birds, one hypothesis is that it navigating via GPS or magnetism. I guess your virtual track suggests that visual orientation is important, but that excludes the influence of if you rotate the box.
Yeah. No, so you don't need vision to maintain a grid pattern. So the rats can do it perfectly well in darkness too, if they have a reference point. So let's say they start out in a certain corner of the box and then they walk around. You get the grid pattern there as well. But without visual input, it's much less stable because they have fewer opportunities for correcting the grid pattern, so that you accumulate error. But the reason is that the most important sensory input for the grid pattern is self-motion, it's proprioceptive inputs. So those are used to calculate the position of where the cell should fire, much more than vision. So vision has rather a role in correcting or calibrating so that you don't get too much drift based on the self-motion. Another thing I should say there is that the grid cells, all that we and others have studied in grid cells is just for short range navigation in the order of a few meters. So when it comes to bird navigation and magnetism, this is long distance navigation, and that is very likely to apply also completely different principles in addition, at least.
Okay, thanks so very much.